Okay, so hello everyone. I'm very happy to be here today, and I followed this seminar series quite a bit and listened to many nice talks. So it's really nice to be able to present here today. So I will present you our work on bacterial rheotaxis in bark and at surfaces. And this is a collaborative project with experiments that have been done in our group in Paris at the ESPCI, together with my colleague Eric Clément. The experiments have been performed by several students and an exchange professor. So the exchange professor is Wanling Ying, who is now at the Northwest University in Xi'an, China. Gaspard Junot is a PhD student who is now doing a postdoc in Barcelona. And Nuris Figuera Morales did also a PhD with us and is now an assistant professor in the Colorado Boulder. And it's a collaborative project that has been done together with Andreas and Arnold, who both did the theory and the numerical simulation for our work. Andreas is now in Vienna and Arnold at the University at UPenn. So let me start with the object that all of this is about. So I will be talking about E. coli bacteria. I'm quite sure that all of you know E. coli bacteria very well. The film that you see here is actually an E. coli bacteria swimming with the body colored in green and the flagella bundle colored in red. And you can see that by rotating the bundle, the bacteria propulses itself. And from time to time, one flagella actually goes out of the bundle. And this happens when one of the motors turns the other way around and opens the bundle. And then this leads to a so-called tumble where the bacteria will change swimming direction. So when these bacteria are put in interaction with flows, a number of very interesting and maybe surprising observations have been made. And I just said two of them here. One of them is that actually bacteria have a tendency to orient upstream and to swim against the flow. And also another one, which is called rheotaxis. This means that bacteria orient with certain directions with respect to the flow. So these observations have been made already, but we would like to look in these effects in a little bit more detail in this talk. So first of all, what I will be doing is to consider the E. coli bacteria and the flow as a fluid structure interaction problem. So let me first discuss a little bit the complex particle, which is actually the E. coli bacteria, which is really a very complex particle. So let me just turn on my pointer here. So in a first respect, you could say that it's an elongated object, a bit like a fiber. But then if we look in more detail, the shape is much more complex. It's formed by a head and a flagella bundle. The bundle has chirality. There is also this bundling and unbundling effect that happens due to the run and tumble motion. Then, of course, it's an active particle. Active particle means a number of things, that there's always a flow perturbation around the object, even if the fluid is at rest. Then also it has a swimming velocity. If I orient it into a certain direction, it will actually propulse itself along this axis. The body rotates, the flagella bundle rotates in the other direction. It can perform run and tumble. And then, of course, it's a small object. It's on a micrometer scale, so we'll always have noise that plays a very important role in our system. This can be just Brownell rotational noise, but also, of course, the run and tumble. And then the last question, there's also some flexibility in the system, which is often not treated a lot, which is mainly localized in the hook, which links the flagella bundle to the body. And the question we will ask now is, I will put this particle kind of as a model particle, which is very well defined in interaction with the flow, and I will investigate what happens. So what we will do is that we will always work with Posay flows because we work with microfluidic geometries in the lab. And we will look at these individual bacteria either in the bulk or close to the surfaces. Now, the question can be asked, how do we observe these objects? So there's two different approaches to observe bacteria under flow. So either we can look at statistical properties. So looking at a statistical property means I would just take a slice in my channel. I will observe in the slice, and I have average, so I have trajectories. But it means I have projections of these trajectories into the plane, and they will be rather short. Or I can have the approach to look at individual trajectories, but keep in mind we are transported under flow and the bacteria are also active on themselves. So we need to do some kind of tracking to be able to follow the bacteria. So for this, you need a Lagrangian tracking. So let me introduce to you the setup that we have built in the lab together with an engineer, with Thierry Darnige, which is a 3D Lagrangian tracker. So it means that by moving the microscope stage, we would keep the bacteria always in focus. So this is very easy if you look at X and Y. So basically you do an image treatment and you assure yourself that your object stays more or less in the center of the image. It is more tricky if you do this in Z. So to track in Z, basically what we do is that we keep the object in focus. So typically you need two images that you need to compare to see if your focus has gotten better or worse by moving in a certain direction. But combining all of this, in the end, we obtain a very good tracking performance. And I have a first film here just to show you how it looks like. So we are tracking this bacteria here. So only the body of the bacteria is fluorescently labeled. You can actually see that here is a microfluidic channel. It actually swims at the surface. So many of you maybe know that bacterial surfaces swim in circles. So that's what we can see. At some stage, the bacteria will actually throw itself into the bulk. So you have also seen that all these other bacteria that were around that we saw before were actually bacteria that were sticking to the surface. They are all gone. Now we are approaching the bottom surface and we can actually see that again, there's bacteria sticking at the bottom surface, but we can still manage to track these bacteria. So you see that the tracking algorithm, this was without flow, works very nicely, but we can actually also turn on the flow and still track the bacteria. I should also mention that for this experiment, there's bacteria sticking to the surfaces, but typically we treat our channels in such a way that there's no bacteria that stick to the surfaces. So this algorithm works very nicely, but we can actually not observe the bacteria body and the flagella at the same time because it's a very elongated object. So if you imagine you want to do image processing and you have either something that is aligned like this or something that you see in a projection like that, the image will be very different. So we are limited to things where we can only color the body. But to overcome this, we actually collaborated with a group in the University of Edinburgh who developed bacteria that have these nice colors that you just saw, where the body is green and the flagella are red. And then we actually use a beam splitter so that we project the body on one part of the camera and the flagella on another part of the camera. And by doing this, we use our tracking algorithm only on the body. And once we have tracked, we add the flagella again. And this was done and leads to the nice film that you have seen here. So with our algorithm, now we can either only visualize the body and track, but we can also track something where we have full access to the flagella dynamics during a 3D trajectory. So let me start with looking at the individual trajectory of an E. coli bacteria transported in a Poiseuille flow. So I think most of you are familiar with what happens if I put an elongated object like a fiber in a shear flow. So the object will, on the one hand, will tumble, and this is just introduced by the rotational component of the shear flow. And at the same time, its center of mass will be transported downstream, but the center of mass will fully follow the streamlines. So this is called a Jeffrey orbit, and it follows these equations that I will come back to a bit later. But now, if I actually use not a passive object, but an active object. So now the orientation becomes extremely important because if I have a certain orientation, the active velocity of the bacteria will need to transport in this direction. So orientation is not something passive anymore, but the orientation of the object will completely determine the trajectory that the object will have. And you have like a loop that I have drawn here, just to imagine that if I displace myself along my orientation, my trajectories will get much more complex or interesting compared to a passive fiber that would just follow the streamlines. And this has been baptized to be an active Jeffrey orbit. So I have actually presented two results here. So this is a theoretical result that has been obtained by Andreas Zöttl and Holger Stark already quite some time ago, where they predicted interesting dynamics from this combination of the Jeffrey orbit and activity. And the first experimental observation was done by the Stocker group, and you can see it here, but it was a projection of a very short trajectory in a certain plane. So we wanted to use actually our tracking algorithm to get more information on the active Jeffrey orbits. So here's our setup, and it's more or less the same setup that you will see overall the experiments that I will present. It's a very classical microfluidic channel. We have a bacterial suspension that is relatively dilute. So we are always in a regime where we are quite far away from any kind of collective motion, and we consider the bacteria to be that individual objects transported under flow. It's injected into the channel, and we actually observe through an objective from here. We work with the typical type of E. coli bacteria. Sometimes they're smooth swimmers, sometimes they do run and tumble. And for most of the situations, we color only the body, but sometimes we color body and flagella. And I just gave typical swimming speeds here and typical sizes of the object. So how we do our observations is that we use, I'm sorry, not in this case. First of all, let me just introduce to you the flow field. So we have checked that the flow field is indeed a Poiseuille flow field that you can see here. So the Z is the height of the channel. And to measure this kind of results, what we do is that we use a high magnification objective and we just scan through the channel so that we have the local velocities at each stage of the channel. So now we use our tracking algorithm and let's see what is the bacteria trajectory in this kind of Poiseuille flow. And you see first by the typical trajectory that is actually shown here. What happens is that the bacteria will actually detach from the surface. So in the experiments, there's a high probability that bacteria are at surfaces. So a lot of trajectories that we observe actually start at the surface. So the bacteria will be detached from the surface and it's in the bottom part of the Poiseuille flow. So there will be a certain rotation, which I actually indicated here, which will turn the bacteria around. So while the bacteria is turned around, it swims. And then something interesting happens, it crosses the midplane. And once it crosses the midplane, the rotation direction of the shear flow has changed. So this bacteria will be oriented back and they will nearly reach the surface again. And then everything starts again. And this leads to these looping trajectories between the two surfaces. So it's the combination between the Jeffrey orbit, the swimming, and the fact that in the Poiseuille flow, we change sign when we cross the midplane. So we can look at this now in a bit more detail and actually try to capture different types of trajectories. And that is what is shown here. So if you start at the surface, well, most of the time you would just end at the surface again. And if you actually don't start in the good angle, you would just leave the surface and will be captured by the surface right away. These orange trajectories are more or less the trajectories that I showed you on the slide before. But the interesting ones are the blue ones where you actually have these swinging and tumbling trajectories. These light blue ones are actually trajectories that indeed cross the midplane and go back and forth. And this dark blue one is one that actually does these nice loops by staying on one side. So we could also simulate with a very simple model, just using Jeffrey's equation and putting activity on top, these type of trajectories, which are the same trajectories that have been presented in the paper by Zettel and Stark before. So with our observations, we could actually recover the observations that have been made for these active Jeffrey orbits. To reproduce them, it's actually enough to use Jeffrey dynamics plus swimming. However, I have to say that these are very short trajectories. If we wanted to look at more loops, of course, noise gets important at some stage. And also something I will come back to later is the rheotaxis. So that these trajectories that are nicely in one plane, so we decided to observe in this plane, would be disturbed if we would like to stay for too long. So just going a bit to like the idea of this 3D, because one of the things that we can do with our tracking algorithm is also look in 3 dimensions. So first here, I have actually a trajectory that has been generated by the numerical code, and I will just zoom through it. And what you can see now is actually we will be looking from above. And how can you imagine these nice oscillatory trajectories? So I showed you for the moment the Jeffrey orbit just in one plane. So if the Jeffrey orbit is in one plane, the object will just nicely rotate around. But if I'm slightly out of plane, and I will come back to that a bit later, I have this kayaking motion. And that's exactly what you see here. The bacteria swims a bit to the side, but on the same time does this Jeffrey orbit. And just to tell you, so this is a comparison between experiments and numerical simulations, and actually also in 3D, it works quite nicely. But now, so this was the idea to look at a single trajectory. Now, if we want to look at statistics and also maybe dynamics that take a little bit longer, we also looked at bulk, but looking at many trajectories. So, well, that's what I just said. And in this case, we're able actually to look at this rheotactic behavior. So the bacterial rheotaxis for the E. coli bacteria has actually been discovered by the Stocker group. And this is the seminal paper by Marcos et al., where they could show that if I put E. coli bacteria under a shear flow, that they will not follow the streamlines, will be just transported downstream, but they have a tendency to be oriented perpendicular to the streamlines. And they could actually explain this very nicely by looking at the shape or at the geometry of E. coli bacteria. So you have the body and you have here this helical flagella. And they have actually shown in a paper before that if you have a helical object due to the chirality of the object and the interaction of the slender body with shear flow, they could predict that actually if you put a helix in a shear flow, the helix as a whole will drift perpendicular to the flow direction. So now imagine you will combine the helix and the body. The helix wants to drift perpendicular with the flow. So here the flow goes from top to bottom, but it cannot really do that because it has the body, and the body is actually much bigger. It has much more viscous friction. So instead of a pure drift, it will actually lead to a torque. So if I have a shape with a helical flagella and a body in a shear flow, it will lead to a reorienting torque that wants to orient the bacteria perpendicular with the flow. And the interesting thing now is once I have oriented it perpendicular to the flow, it will also swim in this direction. So I will have a displacement in this direction. So this was known, but we wanted to look into this into a bit more detail to understand really the mechanism behind it and also to establish some scaling laws. So it's the same setup that I told you about already. And now what we will do again is that we move through slices of the channel with actually our microscope and analyze on each of these slices what would happen. So now here's a snapshot where I'm close to the top and one where I'm close to the bottom. And we have bacteria trajectories that we follow. And I hope you can see and are convinced that these trajectories are not parallel to the flow direction, but they have a bias to the top. So kind of to the left and to the right when you look on the bottom, which is an agreement which has been predicted by the rheotaxis, also by the Stocker group, and of course comes again from the fact that the sign of the Poiseuille flow, the shear rate, is not the same on top and on the bottom half of the Poiseuille flow. So now using those, we can actually make averages about this drift velocity. And this is what is shown here. So what is shown here is actually different positions in the channel. 0.5 is the mid channel, so there's a zero shear rate, so actually nothing happens. And when I get closer to the walls, top and bottom wall, I will increase the shear rate. I can also, of course, increase the shear rate by increasing the flow rate, which is done here. So the first observation is that I will actually increase this drift velocity. So the velocity component that is shown here is only the component perpendicular to the flow. So I can see that this increases when I get closer to the wall, and it also increases when I increase the flow rate, which all of this makes sense. I should point out that we also put passive tracer particles in the flow, and this turned out to be extremely important because later on I will show you bacteria orientations, and we need to be absolutely sure that we have oriented our channel and our observation into the right direction. So it was crucial that we were tracking bacteria and passive particles at the same time. And the good thing is that you can also see here that indeed passive particles do not show this drift velocity. So it's not a default in our channel. It really comes from the rheotactic behavior of the bacteria. So obviously the first thing that you would like to do here is to rescale everything with the shear rate. And it actually turns out to work very nicely. So we did also two things. So we varied the shear rate by staying at the same position and changing the flow rate, or by keeping the flow rate constant by moving through the channel. And you see that clearly the shear rate is the main control parameter that all our data more or less falls on a master curve. And there's also some results from simulation, which I can come back to later again. So two important informations that can be observations can be made here. That actually I have some kind of a linear increase in the beginning, and I have a saturation of the drift velocity. So this drift velocity is normalized by the swimming velocity of the bacteria. The simple explanation for the rheotaxis that I gave to you before actually says there's a torque. The torque wants to orient the bacteria perpendicular to the flow direction. So first of all, it's not completely clear how to understand the linear increase here in the beginning. And also it's not so obvious why it saturates at a value which is really quite below 1, because if it would be perfectly perpendicular, the drift velocity should be proportional or equal to the swimming velocity. So starting from this, we wanted to understand a bit more what was happening here. So first of all, these are average velocities. We expect that they come from some kind of distribution. So first of all, we looked at the velocity distributions for different shear rates. And what you can actually see here is that, of course, no shear, the velocity distribution is very large. So no orientation into a certain direction. And we can see that when we tune the shear rate up, so if we get to more and more and higher shear rates, that we will actually shift the velocity towards, well, one side. So it's actually the orientation to one direction. And we can also see that this peak of the drift velocity gets smaller, but even at very high shear rates, it's not really small. It always stays kind of a large distribution. And we actually combine that with a distribution of the orientations, because if you believe in what I told you before, is that actually the passive orientation resulting from the interaction of the shape and the flow really determines everything that happens here, and that just adds the activity kind of on top of everything. And you can see here on these orientation distributions that if I have no shear flow, the distribution is completely flat. And then I turn on the shear flow, and you can actually see that slowly things move towards the right, but we don't have a single peak, but we always have a double peak. So this double peak is actually, so if this is perpendicular to the flow direction, I'm a little bit above and a little bit below the flow direction, okay? Then of course, if I translate this into a swimming direction, I mean, it's the same flow speed if I'm a little bit above or a little bit below. So I only have one peak here on the velocity. So now the question we ask ourselves, how can I understand these orientation distribution? So let me go back one step and talk again a little bit about the Jeffrey orbit. So I already introduced the Jeffrey orbit, but now I actually have this small animation here. Is actually a Jeffrey orbit in 3D. It's what I showed with my pen before, is this kayaking motion, okay? So what you can see here, and let me maybe just try to play that again. You have a fiber performing this kayaking motion. What is on the right side will always stay on the right side. What is on the left side will always stay on the left side. It's the blue and the red here. And I have presented also the same thing in a phase space, representing the two different angles. So you would cross from right to left here, and you can actually see that you have your small, nicely well-defined Jeffrey orbit on one side. So now Andreas and Arnold have actually added a rheotactic term to these equations, and the rheotactic strength is this new here. And what is maybe interesting to note right away, that the Jeffrey orbit as well as the rheotactic term are both proportional to the shear rate. So basically the shear rate only enters as something that would give the speed which would go around a certain orbit, but will not change the shape of these orbits. So now if I add this rheotactic term, you can actually see that if I start on one side with my head, let's now say, that actually you can see that it's not a stable position, so that actually will spiral outwards. And now it will change sides. So now the head has switched on the other side, and you will see that it will spiral inwards again, and to be aligned as much as possible with a direction close to perpendicular to the flow direction. So if I add the rheotactic term, I completely change the dynamics of the Jeffrey orbit. And so first of all, I break the symmetry. So what is, if I have the head on the wrong side, it would actually switch and go on the other side. And also I really see how I spiral outwards and then again towards this perpendicular direction on the other side. So this is a simulation without noise, but we know that we always have noise in the experiments. And we will add the noise in the next step. But before doing that, I actually should tell you how we actually match the parameters in the simulations and in the experiments. Because you have these rheotactic strengths here, and this G is the parameter of the Jeffrey dynamics, the Jeffrey constant, which is dependent on the aspect ratio of your object. And what we actually did with the simulations is that we used numerical results and the experimental results, here, to actually determine the parameters. So using this curve, we actually fitted the rheotactic strength and the aspect ratio in our model. But the values that we obtained were not random values, but they were actually in quite nice agreement with results from resistive force theory and also previous studies. And the rotational diffusion, we just took it from literature. And once we adjusted these parameters in the equations, this led to this nice agreement that you already saw between our orientation distributions and the experimental results. But now, once we have matched that, we wanted to come back to the question to really understand what is the mechanism behind this rheotactic behavior we observe. And you need to bear with me. This is, I think, the fullest slide of my presentation. So what we presented here are actually two things from the numerical simulations. First of all, the dynamics for a particle that is non-trivial, so simple Jeffrey dynamics, and then what happens for a trivial particle. If I have very strong shear rate, I'm in the situation where I have nearly no noise. And if I have a very small shear rate, I'm in a situation where actually the noise is very important. So let's look first of all at the situation where the noise is very important. So if the noise is very, oh, and sorry, what is in the background here is again these Jeffrey orbits that you saw already. If the noise is very important, more or less, the probability of finding your fiber pointing to somewhere in these orbits is equal everywhere. So now let's go and keep first of all the non-trivial particle. If I turn on the shear rate, we will find the result, I think that most of you know, that the fiber wants to be mostly oriented with the flow direction, which is actually, this is like the zero flow direction and the other one is the opposite. And you can see that indeed this is the case. Situation is very different if I turn on this trivial term, the rheotactic term. We start from the same situation when noise is very strong, but even if I have a relatively weak shear rate, you see that we actually break the symmetry and that everything will end up on the right side, but the distribution is still very large. And you can also clearly see these two peaks that you saw in the orientation distribution before, but the probability to be is highest. And then if I turn on the shear rate more, and the 1000 as a shear rate is actually something that we absolutely cannot obtain in our microfluidic device. And you can see that actually these two peaks have moved together, but still the distribution stays relatively large. So what we wanted to do now is to understand really what is behind that. And to do that, Andreas has actually linearized the equations around specific points. So first of all, he linearized the equation about the orientation perpendicular to the flow direction. And interestingly, what came out of that is that this point is actually only a marginally stable fixed point. So this means that things will circle around until infinity because timescales diverge when you want to approach it. And this explains that even if I'm at very high shear rates, I will not reach the perfect perpendicular orientation because I'm converging towards a marginally stable point. And the second thing he did is that he actually linearized about these two orientations, which are actually the orientations with a lot of noise. And he could show that these 2 points actually move together linearly with shear rate. And this is actually what leads to the linear increase of the rheotactic velocity in the beginning. So by looking at these orientation distributions and then linearizing about certain points, we can completely understand the dynamics of the system. And this is actually represented here. So we have also learned that our control parameter is, of course, the shear rate. Here the nu is this rheotactic strength. And everything is normalized by the noise, the rotational diffusion. So something like a Peckley number and a rheotactic strength or something like that. And with this, we can actually see that in the beginning, I have this linear increase. And then I can have actually these convergence towards the perpendicular orientation, but which is very, very slow. So with this, we think we have completely understood the rheotactic dynamics in the bulk. And in the next step, we will actually go again at looking at individual trajectories, but now close to a boundary. So let's take a step back. So now we still, of course, have all the effects in play that we had for the bulk rheotaxis, but now we add the surface. And what is shown here is using, again, the tracking algorithm. And using the tracking algorithm, we actually increased the shear rate. I think there's a number of things that you will recognize here. If I'm at very small shear rates, I have these circles that we know from bacteria swimming close to surfaces. Then when we turn on the flow, what you can see here is that bacteria are oriented upstream. And if we increase the flow even stronger, I mean, the flow is too strong, so the transport of the bacteria in the end will go downstream again. You clearly see a bias towards one direction, but for even higher flow, we can again observe the trajectories into both directions. So we wanted to analyze these observations in more detail. And again, we came back to the theory. So this, well, maybe I did promise too early that it was the fullest slide, because this slide is actually quite full too. But now we want to take into account kind of everything we can. We want to take into account the fact that the bacteria are elongated objects, that they have chirality, that they have a head. Also, that we have actually interactions of the bacteria with the surface that come from two things. They come from the dipolar perturbation of the flow that will actually lead to an attraction of the bacteria to the surface. But we also have the fact that the head rotates in one direction and the flagella rotates in another direction. So yeah, this is the part I already talked about, sorry. But let me come into detail about these interactions. So first of all, with the wall, we have the interaction that stems from the dipole that would actually align bacteria with the surface and will create some attraction of the bacteria with the surface. Then we have the fact that the head rotates in a certain direction, leading to this swimming in circles that we all know well, I think. The interaction with the bulk, I already described it quite a bit. And then we have another interaction when I have flow and surface, which we call the Weservane effect, and which is again an effect where actually the head will act somehow like an anchor. So when I have actually flow and I turn on the flow, the head will act somehow like an anchor and the flagella will be oriented behind the head. Okay. So now we want to take into account all of these effects. And what is interesting to note is that these wall interaction effects will always be there, even without flow. However, then these bulk and surface interactions are proportional to the flow and it will increase with the shear rate. So we can expect that the dynamics will change with increase of the shear rate. And I just summarized, it's the same approach as before. There will be reorientation rates that are defined, and I didn't write out of the equations. They will be defined by all of these different interactions. And once I have with these reorientation rates found mean orientations, I will add the swimming speed to reconstruct the trajectories. And again, in the simulations, Brownian noise is taken into account. So by using this as a model system, Andreas and Arnold could indeed reproduce these type of trajectories that we saw in the experiments: circling, upstream swimming, oscillations, and also this left and right orientation. So the question is now, where does all of this come from? So let me just in some words explain you with my hands where this comes from, and then we will look at the model again. I think the circling, we all know, the circling just comes from the fact that we have a rotation in a certain direction of the head. Now, if on top of that, I will add this Weservane effect. So the effect that will actually orient the body in one direction and the flagella behind. I will actually go out of the circle to something that will first bring me towards the right. And that's what you can actually say here. And then if I increase further the shear, it will turn myself upstream. So that's the first transition. It's called an Adler transition. It has actually been observed for sperm cells and other objects also. Then if I increase the flow rate even further, I would actually not be able to swim against the flow anymore. It would turn my trajectories downstream. And there's something that we'll come back to where we discovered these kind of oscillatory trajectories that have not been mentioned before. And then at some stage, I break again the symmetry and they can actually swim right and left. So let me move through this a little bit more from the model side. So the nice thing about the model that Andrea and Arnold actually put into place is that they were able to write all the different terms in an analytic way. So that means that they could actually analyze the equations and, for example, try to predict the mean orientation of bacteria from the equations. And that is actually what is done here. So this is the orientation and this is the shear rate. If I'm below this first Adler transition, I would just swim in circles. So this leads actually to this value. And then once I turn on the flow, I will start to orient myself towards the right and upstream, up to a certain optimal shear rate where I'm optimally upstream. And then actually I will bend again slowly downstream and I swim to the right and after a certain transition to the left. And they could analytically determine all these transitions from the equations. Another thing that you can get from the equations is actually the fact that you have an optimal shear rate for upstream swimming, which is given here. And just to mention that we, of course, don't have so many points from the experiments, but if we take from the experiment the mean orientation, they're actually in quite good agreement with what you just saw here. And I just want to mention on a very quick note that right now the angle I showed is actually the angle of orientation in the plane. But we know that actually the bacteria have what we call a dip angle. So they're not perfectly parallel with the surface, but they prefer to have the head a little bit down. And we actually input as the dip angle minus 10 degrees. And while we go through the higher and higher shear rates, this dip angle becomes flatter and flatter. So that could actually be a very interesting information if one wants to know how stable the swimming at the surface is and if a bacteria might detach, for example, from the surface. But I will not go into more detail about this. But I wanted to come back to these oscillations that we observed. So from the trajectories, you could see actually that the trajectories were not straight, but had these oscillations. It probably doesn't really come as a surprise because we talked about the Jeffrey orbit quite a bit already. So actually, in this case, it's a combination between the Jeffrey orbit and the Weathervane effect. So the Jeffrey orbit will always rotate my bacteria around, but here, depending on what half of the orbit I am, I will have actually the Weathervane effect that will come on top of it and will actually help the orientation. So to see if this mechanism is really also there in the experiments, we came back to the experiments and actually looked more carefully. And we also did some experiments where we actually labeled the bacteria fluorescently with the head and the flagella. And that's what you can actually see here. So you can clearly see that the orientation of the flagella will actually change over time. And we can actually, from this orientation, measure the frequency. And we also looked at velocity fluctuations for the trajectories that you saw before. And when we combine all of this, we can actually get this oscillation frequency diagram. So the first part is actually when we are still swimming in circles, so nothing can be seen here. And then we have this onset of oscillation frequencies once I have actually crossed the transition to oscillatory frequencies. And this yellow line is actually the prediction from the theory, which is mostly given by the Jeffrey frequency, but actually all the individual terms play a role here. So this is the mechanism. Let me just, as a final point, come back to the distributions. This really summarizes what we've seen on the trajectories. So these are orientation distributions for smooth swimmers, so no run and tumble. And we can clearly see that we go from a very broad distribution to something that will actually go to the right, upstream, and then splitting into right and left. And there are some experimental observations, but the statistics on the experiments are actually not really great on this respect. So with this, we think that we have kind of fully understood the dynamics of bacteria that are transported close to surfaces. Actually, these dynamics are very complex. So I have a lot of different types of mechanisms going from the circular swimming to the oscillatory trajectory until this bifurcation of right and left. So once we know exactly what are the dynamics close to surface, one could also think that maybe that's something that could be linked to prevent upstream contamination or to direct bacteria to certain stages or places in the flow. So the last part of my talk, I actually really want to talk about this question about this upstream contamination. So to do this, we did actually look not only at the surface, but we looked at a corner. So we know that the bacteria, so in general, if you look at transport, or if you want to look at transport of bacteria, it's very interesting to look at surfaces because we know that the concentration of bacterial surfaces is very high compared to the bug. Not really bacteria sticking at surfaces, but just, I mean, there's two things. First of all, you have this hydrodynamic attraction with surfaces, but also just from a dynamical or sterical point of view. I mean, once an object swims and comes close to a surface, it just needs some time to change its orientation if it wants to leave again. So they're really privileged places in flows. And even more though, if you make a corner or if you look at the corner, because also the shear is actually lower at the corner. So what we said, okay, let's start to look what happens at the corners. And we know that upstream swimming happens at certain shear rates. And I just have like this sketch here again. So also when I arrive to a corner, I have the same reorientation mechanism that I introduced as the Weathervane effect before. Just now I have like a 1D corridor where my bacteria will go. So let's see what happens. So the experiment that we have designed is actually the following. So we have a reservoir filled with bacteria. I have a reservoir with clean clean, and they will actually create a counterflow with this stock solution. And then I will actually observe here at the junction between the bacterial reservoir and the clean solution if the bacteria will manage to travel upstream in this channel. So the observations that are shown here, so the bacteria are fluorescent. So you can actually see you have the reservoir, you have the cell-free medium that comes from here, and then they will just travel upstream. And the remarkable thing is that after 13 minutes, bacteria have managed to contaminate this channel by really a macroscopic distance of 13 millimeters. So this upstream contamination is actually more efficient if I turn on the flow than if there was no flow. Because if there were no flow, up and downstream swimming would be equally probable. But if I turn on a little bit of flow, I would actually turn them upstream. So we wanted to understand how the bacteria actually managed to fill the channel and travel upstream. And for this, we actually measured concentration profiles here in the channel. Okay, not yet. To understand how actually the mechanism works, we actually tracked individual bacteria at the walls of the channel. So what you can see here is a small zoom on a certain part of the channel. You can see the bacteria actually swim upstream on the wall, and from time to time, one of the bacteria will be detached from the wall. And when it's detached from the wall, it will be transported downstream in the flow until it manages actually to attach again on another wall. So sometimes you see that they go from this one, for example, from one wall to the other wall, but often they actually detach and then transported downstream. And that's also what you can actually observe here. They swim upstream, transported downstream, they swim upstream, transported downstream, etc. So this is the mechanism of contamination. At the edges, they swim upstream, and they're detached, they're transported downstream. So we wanted actually to input this in a very simple model, which is just drawn here. But the interesting ingredient here is that we make one hypothesis, which is the fact that they will actually swim upstream until they want to do a tumble. And once the tumble occurs, this will actually lead to detachment of the bacteria from the surface. So when we did this study, we didn't have really evidence of that yet, but now we have these observations with the bacteria with the head and the flagella colored, and we have direct evidence that detachment is linked to tumbles of the bacteria. So this means that we actually inject tumbling statistics into our model to get the modeling result. And now finally, I have what I already said before. So actually what we measure is that you have these concentration profiles. It's contamination profiles. So how far have the bacteria managed to move upstream in the channel? Then we use our simple toy model, and we can actually get a very nice agreement between the theory and the experiments. So what is interesting about that is a some number of things. Of course, we see that there's really strong upstream contamination that can actually take place in confined geometries. But I think the second really important point is here that to understand this contamination process, we need to have knowledge about the run and tumble statistics, because it's the tumble that will actually take the bacteria off the wall. And really on a side note, I would just say that even from this, we could see that if we model the run and tumble statistics as a Poisson process, which is the simplest model that has been given by Burke, that actually the statistics don't work well to describe these very, very long tails of the contamination process. And with this, I have actually finished the part on the bacteria. And as an outlook, I wanted to tell you that everything I did right now, or that I described before, was on living bacteria. So we could wonder to what extent my idea that it's only or mainly a fluid structure interaction process that determines the orientation, and on top of that I put the activity, is true. What we actually did in the lab also is that we produced what we call passive bacteria, and we like this term a lot because everybody or a lot of people try to make active colors, so we make passive bacteria. So we actually have a nanoscribe in the lab which can produce tiny objects on the micrometer scale. And we printed small bacteria with a head and a tail, and we put them under flow. And the nice thing now is that we have control of everything. We can change the shape of the head and the change of the flagella. And I don't really have time to talk about it now, but we can actually also with those verify some of the predictions of the rheotaxis that we observe with the bacteria. And with this, I want to tell you that the bacteria are really very fascinating objects, very, very complex particles, but there's still a lot of things to be done. In particular, what is still missing somehow in our picture is how I link the bulk to the surface. And with this, I would like to thank you all, and again, thank my collaborators. And I'm very happy to take any kind of questions that you might have.